and we're off. Okay, good. Okay, folks, let's get started. How's everybody doing today? Good. Extra credit right there to Melissa. If I learn your name in this class, you're in trouble. So that's one of the things to learn here. So, everything making sense? Is anything making sense? I got a yes to that. So something is making sense or everything is making sense. I can pick whichever one I choose, right? Okay, good. All right, so we're making our way through the protein structure, and I hope to finish that today. I may not quite get it done, but I will hope to finish that. That'll be my aim, and we shall see how it goes. So last time I talked about secondary structure. Yes? It's cutting out? Like horrible? Like worse than me? Thanks. All right, well, I'm trying something new with the microphone. Is that a little better? No. Is that a little better? How's that? Almost non-existent? You want me to just turn the microphone off? Yeah. Okay, I got no problem with that. I got a big mouth. One less thing to wear. If you can't hear me up there, you can't hear me, give me a sign, like maybe lightning bolt or something, okay? Good. I'll call you God if you do that, okay? Okay, so we'll test my vocal cords today. I'll spit all over the first row. Anyway, last time I talked about secondary structure of proteins. I have one last thing to finish up regarding secondary structure proteins that I want to talk about, and then we'll talk about tertiary, quaternary, and other considerations about proteins. So the one last thing I want to talk about with respect to proteins are a subclass of proteins. So far we haven't talked about individual proteins. But now I want to say something about a special group of proteins. And this special group of proteins is notable because they are what we describe as fibrous proteins. Okay? Fibrous. As their name would suggest, they exist mostly in a fibrous form. And they generally don't have much besides primary and secondary structure. That is, they don't really have much in the way of tertiary structure. They mostly exist as polypeptide chains that have secondary structure. That may be beta strands arranged in sheets. That may be alpha helices, etc. In some cases, the alpha helices may be coiled, kind of like you see here. This class of proteins is not the largest class of proteins, okay? By a long ways. This is a relatively small class of proteins, but it's an important one, especially for a bald guy like me, because this includes hair. Hair is a fibrous protein. Okay? So fibrous proteins, as I said, don't really have much in the way of primary, in the way of tertiary or quaternary structure. They have pretty much primary and secondary, and that's about it. Now, as you might expect, they're fibrous. They go on and on. Your hair is a good example. Silk is a good example. And silk is a good example of a fibrous protein that has beta strands arranged in sheets. Okay? Real good example of that. Your nails, your fingernails, claws. Okay? These have a lot of fibrous protein. Teeth have a lot of fibrous protein associated with them. And as you can see, fibrous protein can give hardness. They can give strength and a variety of qualities to a substance. Okay. A very interesting fibrous protein is that of collagen. Collagen is the most abundant protein in your body. It's literally the glue that sticks you together. Without collagen, you're not sitting here. You are a lump on the floor. You're a puddle of cells. Collagen helps hold us together. Okay? Now, collagen, as I said, is a fibrous protein. Let's look at the sequence. This is the primary structure of collagen. And when we look at this, we see something very unusual. A lot of prolines. Proline, proline, proline. That HYP stands for hydroxyproline. Proline, hydroxyproline. Look at that. Now, I've already been telling you that proline is this oddball that causes bends, that causes problems, etc., with the structure of, say, a helix. But here's a helical protein, and it's full of proline. Well, I think you can begin to quickly see why this is able to get away with being a fibrous protein. Okay? Proline doesn't have much flexibility, but look at the amino acid that's next to it. Glycine. It's got the smallest R group. It has absolutely the most flexibility that we can have. Okay? So glycine is able to compensate for some of the limitations of proline. And that's why I tell you we can't look at any one individual amino acid and decide if is this amino acid in a secondary structure or not, like an alpha helix, because it kind of depends on what it's around. So this is a prime example, collagen. Now, hydroxyproline we haven't talked about. And I want to just say a word about this, especially for anybody interested in medicine. So this is a very, very interesting modified amino acid. When we examine the sequence of collagen, what we discover is that collagen actually has 2 amino acids that get modified after the protein is made. So the original collagen molecule is made with proline. It's also made with lysine. But both of these amino acids get modified in a chemical reaction frequently, as you can see on the screen. They frequently get modified to add a hydroxyl group to them to make hydroxyproline and or hydroxylation. Well, why do we care about this from a health perspective? Well, from a health perspective, it's a very interesting thing. Oop, not what I wanted. Not what I wanted either, although that's getting there. Well, I thought I had another structure. Okay, let me just go back to this one. All right. Collagen is a helix of helices. 3 helices put together to make a bigger helix. I want you to think about braiding of your hair. Okay? The braiding of your hair, okay, is something that gives that braid greater strength than any of the individual strands comprising it. Right? So braiding, combining things, actually helps to build strength, and that's one of the ways in which collagen itself remains fairly strong. Gesundheit. Okay? Why the hydroxylation? Well, the hydroxylation, it turns out that when you put hydroxyl groups pretty close to each other in the structure of a collagen protein, they will frequently react with each other and split out water to make a covalent bond between them. So let's say this red guy has a hydroxyl right here, and the blue guy has a hydroxyl right there. These two combine and make a covalent bond. And covalent bonds, you remember, are the strongest bonds. Not only are you braiding the hair, but you're putting a rubber band in at the bottom so it can't come undone. If you don't put something to hold the hair together, the braid all comes apart, right? So what this modification allows is a covalent bond to help strengthen and hold together these strands. As a result, the collagen not only is strong, but it remains strong. It doesn't come apart. Okay? Now, well, what happens if we don't make those modifications? If we don't make those modifications, we end up with a collagen that is very, very weak. Okay? Very, very weak. You literally start falling apart because your collagen isn't any good. The story I like to tell is that the way this was discovered was actually back in the days of the pirates. True story. The pirates used to take off and go off on these giant conquer the world expeditions where they'd get on a boat and they would be there with a bunch of stinky other guys, great big burly pirates going out here. The main thing they ate was salted meat. They didn't have refrigeration. They didn't have quiche. They didn't have a variety of things. They ate salted meat. That was basically what they ate. Okay? And these pirates would go out as big hulking guys, and they came back as little baby wimps, falling apart. They were unable to modify their collagen. Why were they unable to modify their collagen? Well, it turns out that the thing that's necessary for modifying the collagen is vitamin C. Vitamin C is necessary for that reaction. If you can't modify your collagen by having no vitamin C, you develop a condition known as scurvy. Scurvy is not something you want to encounter in your lifetime. Today's world, modern industrial societies, scurvy is almost non-existent because people get fruits, vegetables, and things besides salted meat in their diet. Meat doesn't contain an awful lot of vitamin C, and that's why the pirates developed scurvy. Make sense? Clear as mud? I kind of like this shouting thing. I can just belt it right out here. This is good. Yes, ma'am.
So for collagen, with the autoimmune diseases, we have an excess production of collagen.
Yes?
There's just more strands that are being braided in.
I'm sorry. Say the first part again.
So, like in an autoimmune disease, like scleroderma or something, when the body produces an excess amount of collagen around the tissues, does that just mean that more strands are being braided in?
To be honest, I don't know. I would imagine in an autoimmune disease, the problem that you have are actually related more to the immunity than they are to the production of additional things. But I don't know about that particular disease, to be honest with you. I'm sorry. Autoimmune diseases are probably even worse than scurvy. You probably don't want to have to mess with them either. They're not much fun. What's that?
They're weird.
They're weird. They are weird, yeah. And they manifest themselves in many very different ways. Okay, yes? Lysine and proline. They both get hydroxyl groups, and those hydroxyl groups come from vitamin C. In fact, this is the only biochemical reaction, okay, I'm aware of where the vitamin C actually is a substrate in the reaction. It's actually a substrate. It actually does something in this reaction. Most vitamins are coenzymes or cofactors that help enzymes to do their things. But in this case, vitamin C actually is a substrate. It's necessary for the reaction. Okay, well, let's turn our attention now to tertiary structure. So primary structure was the sequence of amino acids. Secondary structure was the regular repeating structures that we saw that arose from interactions between amino acids close in primary sequence. Now we turn our attention to tertiary structure, and I'm going to give you a definition here as well. Tertiary structure is a structure of proteins that arises as a result of interactions between amino acids, and yes, I'll repeat this, that are not close in primary sequence. So tertiary structure is a protein structure that arises from interactions between amino acids that are not close in primary sequence. When I described what close meant in the in the secondary structures, I said within about 10 amino acids. That's what we're talking about for interactions that are close to each other. Now we're talking about interactions between amino acids that are farther apart than 10 in the primary sequence. So let me show you a real good example. It's a classic example, and it is that of myoglobin. Myoglobin is a protein that has primarily alpha helices. You can see the alpha helices in this structure. And you can see how this thing goes, and it bends, and then it gets another helix, and it bends, and it gets another helix, and it bends, and another helix, and it bends. Kind of like what we talked about. All right? Tertiary structure gives three-dimensionality to proteins. In order for us to have tertiary structure, we have to have these bends that I was talking about. Okay? We have to have those bends in order to have tertiary structure. Otherwise, we have simply a fibrous protein. Right? So these bends are necessary to have that overall structure. Because of these bends, we may have something like an amino acid, let's say right here where my pointer is, that's amino acid number 21, that is in very close proximity to this amino acid over here that might be number 91. These guys might form a hydrogen bond. As a consequence, we have interactions because of the bending between two things that are not close in primary sequence but are close in three-dimensional structure. The bending brings together things that were not previously close together. If they were in a fibrous form laid out, there's no way 21 is going to be close to 91. But when I bend it, they can be. Okay? So tertiary structure requires those bends, those turns, in order to get things into this proper configuration. Now, tertiary structure gives rise to what we call globular proteins. Globular proteins. Globular proteins are the predominant type of protein that we find in biology. Okay, we find them in the membranes of cells, we find them in the cytoplasm of cells, we find them all over. Enzymes are globular. So probably 99% of the proteins are globular. So you can see the fibrous are relatively minor proteins in terms of numbers of proteins. Collagen, of course, is very abundant, but that's in terms of numbers, globular trumps everything there. Okay, so that's what's happening with those. If I look at this guy right here, when I look at a globular protein, I see several things that can stabilize it. So when I talked about secondary structure, one of the forces that stabilized secondary structure was hydrogen bonds. And I said that was the predominant force that helped to stabilize secondary structures. In tertiary structures, we have many more possibilities for stabilizing forces. And I'm going to give you some of those. Hydrogen bonds are one. Okay? Hydrogen bonds are one. Another that is very easy to understand is that of ionic bonds. If you think about that alpha helix with the R groups pointing outwards, okay, there's not a lot of opportunity for those R groups, a minus over here, to interact with a minus over here. They're not in real close proximity. However, if I fold up a protein, we could imagine that an R group right here might be very close to an R group over here. This might be a carboxyl. This might be an amine group. And if that carboxyl has a minus and the amine has a plus, they're attracted to each other, right? So ionic bonds become much more of a factor in tertiary structure. They could be attractive in nature. They could also be repulsive in nature. Let's imagine that this guy right here is a ways away from that guy right there. They're both positively charged, and that repulsion keeps them a certain distance apart, just like two guys in a bar that want to fight. You keep them far enough apart, they're not going to fight as much, right? So that repulsive distance can also be a factor. What we see now is if we change the charge, which is what we've been talking about with respect to pH, if we change the charge, what's going to happen? What was previously an attractive force may no longer be attractive. What was previously repulsive may no longer be repulsive. And so as a consequence of that, we could imagine that that protein may change its shape at least slightly. In some cases, it might change its shape drastically. Okay, so we have hydrogen bonds. We have ionic bonds. And we have something else that's very interesting. And it's something you have a sort of intuitive feel for. You don't have to understand the thermodynamics of it to understand this third force I'm going to describe. The third force relates to what are called hydrophobic interactions. Now, when we think of hydrophobic interactions, you probably thought of it as, well, that's why oil separates from water. Oil is very nonpolar, and the nonpolar guys like to stick together. They don't want to dissolve in water because water is very polar, and it's like being at a party and you're the shy person and all those people over there interacting and you don't want to interact with them. You want to just go over here in this corner and go die, right? I used to feel like that. I used to be a very shy person at one time, but that was a long time ago, all right? So I know that feeling. That repulsive force is very strong there. I don't want to be there, so I want to associate more with the people that want to die at a party than I do with the people that want to be the social butterflies at the party. So we go and hang out together, right? So in the inside of a protein, we discover something interesting. That's where we find most of the hydrophobic amino acids. At least if that protein spends most of its time in water. So for water-soluble proteins, when we look at their distribution of amino acids, we see that they are associating with each other on the inside of that protein. So they're avoiding water on the outside. They're avoiding social contact. They're very happy with each other. They're very comfortable with each other. And on the outside of the protein, you can guess what's out there, the ones that like water. Okay? So there's an uneven distribution of amino acids when we look at proteins that normally are dissolved in water. Not all proteins are normally dissolved in water, and I'll show you a reversal of what I just told you in a minute. But everybody understands what most proteins are like then, right? Those hydrophobic forces really are reasonably strong when they're added up. They help to give stability to that protein. No, don't make me go talk to those people over here. I'm going to resist that. I'm going to stay here and hold this protein together. Okay? So hydrophobic interactions are a stabilizing force for tertiary structure, but certainly not for secondary structure. Okay, so there's three forces: hydrogen bonds, ionic bonds, and hydrophobic interactions. There's a fourth one, and the fourth one turns out to be the strongest one. Now, this one doesn't really happen in secondary structure, although there are a couple places where it appears, and I may even show you one of those. But for the most part, we think of this fourth force as happening only in tertiary and quaternary structure. Let me describe it to you. Let's say again, we've got this folded protein that's up here. Let's say I have a cysteine residue that's right there, and sticking off of that cysteine residue is a sulfhydryl, which is what a cysteine has as its R group. Sulfhydryls turn out to be fairly reactive. They're fairly reactive. Imagine, if you will, that not only do I have a cysteine with an SH right there, but I have a cysteine with an SH right there. One of the ways that cysteine sulfhydryls are reactive is if you put two of them close together, they will split out the hydrogens, they will lose their electrons, meaning there's an oxidation that occurs, and you make a disulfide bond. Sulfur directly to sulfur. Okay? A disulfide bond is a covalent bond. It's a very strong bond, and that's why it's the strongest of the four forces that I've just described to you. In order to make a disulfide bond, we basically have to be able to fold something together in order to get those sulfhydryls in close enough proximity so that they can react. There are some minor exceptions to that, but that's basically the rule. Yes?
So can methionine make a disulfide?
Can methionine make a disulfide? Good question. The answer is no, it cannot. Because to make a disulfide bond requires a sulfhydryl, and the sulfur in methionine is joined to two other carbons. So methionine cannot make a disulfide bond. Now we'll talk in a little bit about chemicals that we can use that will break disulfide bonds, and we'll see the significance of those later. But before we think about that, I want you to think about, okay, here I have a disulfide bond. A disulfide bond is very strong. I can't break a disulfide bond by boiling it. I can break a hydrogen bond by boiling it. I can break hydrophobic bonds by putting them in boiling water. I can break hydrophobic bonds by putting some detergent in there. What does detergent do? What's the function of detergent? Why do you wash your hands with soap? What's it doing? Anybody know?
The hydrophilic gills interrupt the hydrophobic heads.
Okay, so you wash your hand with soap because soap has a hydrophobic tail, and it solubilizes hydrophobic molecules in water. So you have grease on your hands. The reason you use soap is that grease, which won't come off with just water, will dissolve in water when you put soap there. It's disrupting hydrophobic interactions. If you put detergent in with a protein that has hydrophobic bonds helping to hold it together, what's going to happen to that protein? Well, the hydrophobic bonds aren't going to be there anymore. The protein is probably going to partly come apart. One of the reasons you wash your hands is for that very reason. The bacteria are full of proteins that you're just denaturing their proteins right there. Oh my God, am I denaturing the proteins in my skin? Yeah, you betcha. You got a lot of skin, folks, and most of the skin that's on your hands is already dead anyway. It's good to kill those bacteria, right? So washing your hands with soap, that's why you do it. That's why that helps to kill bacteria, because you're nailing their proteins right there. Okay, now, those are the four main forces that stabilize a protein. There's a fifth one. We talk about it occasionally. It's not going to talk about it much in this class. It's called a metallic bond. And as you might imagine, a metal ion is involved in helping to stabilize that. We're not going to talk about those here. Okay. So, if we look at this myoglobin that I showed you before, which is a water-soluble protein, and we look at the distribution of amino acids, we can see that there's uneven. Blue being the more polar amino acids that are on the outside, the yellow being the more nonpolar that are on the inside. We've sort of peeled this away so we can see what's on the inside. But you see that uneven distribution that I talked to you about before. There's one other thing in here I haven't mentioned, and we'll be talking about this over the next couple class periods quite a bit, and that's this little group up here that's in black. That's not an amino acid. It's actually another thing that we find in some proteins. It's called a prosthetic group. So a prosthetic group is just like a prosthetic limb, is an artificial component of a protein. In the case of myoglobin, that prosthetic group is known as heme. Heme, you may know, is an iron-containing substance. It's found in hemoglobin. It's also found in myoglobin. And it's involved in carrying oxygen. So if you want a definition of a prosthetic group, it's simply a non-amino acid component of a protein. That's all it is, a non-amino acid component of a protein. Heme, it turns out, is almost identical to chlorophyll. Very, very similar in structure. Okay. Now, I had some exceptions for you. I said if I have proteins that are dissolved in water, I pretty much have, and by the way, you saw in that last structure, there's no absolute thing. All of the hydrophilics are on the outside and all the hydrophobics are on the inside. That's not what happens. Those are general tendencies. But if we look at a protein that's not contained in water, but instead is normally found inside of membranes, we discover something very different. Why? Well, membranes have a nonpolar interior. Okay? You talk about the lipid bilayer. The inside of that bilayer are long tails of fatty acids that have nothing hydrophilic about them. We can imagine that a protein that spends most of its time in the membrane might want to fit into that membrane. So it might want to have on at least some of its outside amino acids some hydrophobic amino acids that can interact with that membrane. And that's exactly what we see. Okay? This guy here is a protein called porin. Porin is interesting in that porin is, first of all, it's a membrane protein, so it's found only in the membranes. We would expect that it would have an unusual distribution of amino acids, and it does. Okay? So we see largely a hydrophobic exterior out here. The function of porin is to let water into the cell, or out of the cell as appropriate. This guy has everything reversed. On the inside, we see the hydrophilic. On the outside, we see the hydrophobic. Why? This guy's not spending most of its time in water. Water's spending most of its time in it. Kind of cool, huh? So, the chemistry that we see associated with proteins reflects the environment in which they're in. It also reflects the functions that the proteins have. And we'll see more about that as we go along. Okay. Here's a disulfide bond. I didn't show you that earlier, but I'll show you here. There we go. You put two cysteines close together, they make a disulfide bond. As I said, that reaction is an oxidation reaction. If we start on the left and we move to the right, we lose protons, we lose electrons, and anytime we lose electrons, that's an oxidation. The product of that reaction is a covalent bond. The covalent bond is the disulfide bond right here. And if we want to go backwards, which we do sometimes, our cells will do that sometimes, what we see when we go backwards is it takes electrons, it takes something capable of donating those electrons in order for that reaction to go backwards to the left. And yes, cells do that in addition to human beings working in a laboratory mucking around with proteins. Okay? We have proteins that get oxidized and get reduced by factors inside of cells. And again, the reactions go either left or right depending upon what the cell is trying to do. Okay. Here's an interesting protein. It's insulin. Okay? Everybody's heard of insulin. And you probably know insulin is the protein that is either deficient or not behaving properly in the bodies of people who have diabetes. We'll talk a little bit more about that later. But we're going to look at insulin from the perspective of structure. This doesn't look like any of the proteins you've seen so far. Insulin, look at it. You see two different polypeptide chains. What holds them together? Why don't they go flying apart? Well, you'll notice what holds them together are two disulfide bonds. Without those disulfide bonds, those chains don't stay together, insulin doesn't function, and you basically die a horrible death. Okay? Now, how does this get this way in the first place? Let me just tell you briefly how this happens. It turns out insulin starts out as a single polypeptide chain. A single polypeptide chain. Then, after it's made, there's a protease that comes along and it clips the end off. So it was sort of folded around there. If you clip that little fold off, what was one polypeptide chain becomes two. Okay? So insulin started out as a single polypeptide chain, but after that clipping, it makes two. We'll see later in the class that clipping, just like I described to you here, clipping, breaking covalent bonds between amino acids, breaking peptide bonds, is sometimes a very important factor in activating proteins. It's definitely the case for insulin. If you don't clip that bond, insulin doesn't have any activity. It doesn't do what it's supposed to do, which is to help regulate blood glucose. So that clipping is necessary in this case to give insulin its proper function. Yes, ma'am? Okay, so I said it was clipped by what? Insulin is clipped by an enzyme called a protease. A protease is a protein that catalyzes a reaction on another protein. The reaction it catalyzes is breaking a peptide bond. Okay, everybody with me? Questions? I don't have any jokes for you today, I'm sorry. Okay. All right, that's what I want to say about tertiary structure. Let me turn our attention briefly to quaternary structure, and then we're going to talk over some examples. So, what's quaternary structure? Well, let me describe it to you. Quaternary structure, we've seen that primary was a sequence of amino acids. Secondary was interactions between amino acids that are close. Tertiary was between amino acids that aren't close. What does that leave for quaternary? It leaves different polypeptides interacting. So you sort of saw that with insulin. I told you it started out as the same one, but you're seeing two separate polypeptide chains that are interacting. That's what quaternary structure is. The best example I can give you is that of hemoglobin. Hemoglobin is related to myoglobin, but it has a fundamental difference. The fundamental difference is instead of having one polypeptide chain, hemoglobin has four. It has two identical copies of one subunit called alpha and two identical copies of another subunit called beta. That's the composition of what we call adult hemoglobin. We'll talk about fetal later. Okay? Those four, and you can see the two alphas, I think, in red and the two betas in yellow. It doesn't really matter for our purposes here. Are held together by the same combination of forces that stabilize tertiary structure. The same combination of forces. So we may have hydrogen bonds involved. We may have ionic bonds involved. We may have metallic bonds involved. We may have hydrophobic interactions involved. And we may have disulfide bonds involved. So all five of those will help to stabilize quaternary structure. All five of those. If you look at hemoglobin, it has the shape almost of a donut. And it turns out that donut shape has a very important property right there in the middle. We'll talk about that very soon and how that relates to people who smoke. Okay? A very important consideration in smoking. Okay, so if we look at the individual components, alpha and beta subunits are fairly closely related to each other. They're not real different from each other in terms of amino acid sequence. They're also fairly close in sequence to myoglobin. They're not that far off of myoglobin. But myoglobin always stays as a monomer, meaning one polypeptide always by itself, whereas hemoglobin always is present as a tetramer. Okay. Because hemoglobin has a very similar structure to myoglobin, it also has hemes, and you see a heme there, a heme there, a heme there, and a heme there. Each subunit has a heme group. And of course with hemoglobin it's no surprise because the name says heme, right? But both hemoglobin and myoglobin have a heme group. That's something that's true of both of those. Both of them are oxygen holding or carrying proteins. We'll see the role of myoglobin later in that respect. Okay. I kind of like this figure. It is a depiction of the coat of a virus that that person who just coughed has. Okay? It's called a rhinovirus. Rhinoviruses cause common colds. There's many things that cause symptoms of common colds, including the flu virus. And by the way, if you haven't gotten your flu shot, get your flu shot. Especially anybody interested in being a health professional really ought to be taking the appropriate steps to protect not only themselves, but helping to stop the spread of disease. So get a flu shot. My strong advice. I got one just yesterday, actually, so it's important to get out there and do that. Okay. This guy right here is what causes a lot of symptoms of the common cold. And I like this figure because it shows in color different proteins that are interacting with each other in quaternary interactions. Here's a green protein interacting with a blue, with a red, etc. This is the outer coat of this virus, and this is one of these self-assembling structures that I told you about. The pieces literally put themselves together to solve the puzzle. What favors that going on? The forces I've been describing to you. Hydrogen bonds, ionic bonds, metallic bonds, hydrophobic interactions, and disulfide bonds. Okay? These things are all helping to drive the self-assembly of something like a cold virus. Pretty cool stuff. Okay. At that point, perhaps I thought we'd take a break and sing a song. Would you guys like to do that? Okay. This is a song I wrote a long time ago. By the way, my rule is if you don't sing loudly, I don't do songs anymore. So if you really don't want to hear songs, just don't sing anything, okay? Then I'll just shut up. What's that? Do I sound like a singer? Oh, she's extra credit. Okay. This one is to the tune of "Oh Little Town." What in the world? That's the wrong one. Sorry, I got the wrong one. This is the tune of "Oh Little Town of Bethlehem." Okay? Oh, we can all sing the whole thing. Oh, little protein molecule, you're lovely and serene, with 20 Zwitter ions like cysteine and alanine. Your secondary structure has pitches and repeats, arranged in alpha helices and beta pleated sheets. The Ramachandran plots are predictions made to try to tell the structures you can have for angles phi and psi. And tertiary structure gives polypeptide zing because of magic that occurs in protein folding. Oops, lost my thing. A folded enzyme's active and starts to catalyze when activators bind into its allosteric sites. We'll talk about that later. Some other mechanisms control the enzyme rates by regulating synthesis and placement of phosphates. And all the regulation that's found inside of cells reminds the students learning it of pathways straight from hell. So here's how to remember the phosphate strategies. They turn the GPBs to A's and GSAs to B's. That last we'll talk about also later, so we'll talk about that. I don't have a thing that covers the thing exactly, so, okay. All right, so let's get back to important things. Now there's one thing that came up in there I haven't really said much about, and I want to spend a little bit of time talking about it, and that is a very, very, very important consideration in protein structure. It's not the structure itself, but how it gets there. Okay? We saw that three-dimensional structure for myoglobin, and we just took for granted that, well, it just happened. It didn't just happen. Okay? It didn't just happen. It had to fold. It had to come together in some appropriate way. What determined how it came together? Well, ultimately, it was the, what was it? Amino acid sequence. The amino acid sequence ultimately determined what was capable of coming together and interacting. Right? So a certain amino acid is going to favor a fold this way versus that way, etc. Folding has to happen in order for a protein to get its three-dimensional structure. Underline that, underline that, underline that. Now I'm going to tell you a statistic about folding that will probably blow you away the way it blows me away. Okay? How does folding happen? What do we know about folding? Well, it ought to be fairly simple if we simply take the sequence of amino acids and we punch it into a computer and we say, what should be the three-dimensional structure of this protein? We ought to be able to predict it pretty well, because we can certainly predict secondary structure fairly well. I told you that. Tertiary structure ought to be a piece of cake. Turns out we don't have a clue in a lot of cases. Okay? It's impossible today for us to take the sequence of a protein, give it a computer, and have the computer tell us the three-dimensional structure. It really is. Why? Well, there's a lot of possibilities. There's an angle here. Okay, we got all these different twisting angles, the phi's and psi's, right? And then we've got individual twists of the R groups. So we've got a lot of different possible configurations that can happen. Now, this is the thing that will astonish you. We say, well, how does folding happen? If we say we look at all these possible different angles and all these different things that can happen in a protein, if we give it into a computer and we tell the computer, okay, try every possible one that can be there, try every possible angle, and we'll just randomly go through it and see how long it takes for the computer to solve the problem. Okay? So we give it to the most powerful computer on Earth, and we give it a protein that's only 50 amino acids long. That's not even a very long protein. 50 amino acids long. And we say, tell us, by using all the different possible structures, find the right one. If we did that, we would never get our answer. We could give it to the most powerful computer on Earth, and it wouldn't be able to give us the answer. We could give that problem to every single computer on the face of the Earth and add all of their processing power together, and it wouldn't be able to give us an answer. In fact, if we gave it to every single computer on the face of the Earth today, the billions of computers that are out there, and we said, work at this at the rate of several trillion calculations per second, or quadrillion, I guess is what we'd have in today's world with the computers that are there, several quadrillion operations per second, it would take 1,000,000 times longer than the age of the universe to go through every possible one. 13 billion million years to solve one 50 amino acid protein. Yeah, this is a computational problem that's somewhat beyond our abilities. And it's unlikely to be one that we're going to solve by brute force, because it's unlikely we're going to make in our lifetimes, a computer that's 100 billion trillion times faster than it currently is. Okay? Now, what does this tell us? It tells us, first of all, by the way, this is called Levinthal's paradox. Okay? Levinthal's paradox. Levinthal's paradox says that there's a hell of a lot of possible structures, but somehow proteins got it all figured out. When you start making a protein, it starts folding. It has that ability, okay? It has that ability chemically built into it. Those chemical reactions, those slight charges here, that slight shape change there is built into it. We just don't fully understand them. But a protein can fold in the order of seconds to minutes. A pretty phenomenal thing. There are things that help proteins to fold. We'll talk a little bit about those in a bit. Things that help proteins to fold are called chaperonins. A chaperonin is a complex that allows a protein to fold properly. Folding properly is really important, because if we don't fold properly, we create a protein that is literally out of control. You've heard of mad cow disease? Mad cow disease arises because of an improperly folded protein. Yes, ma'am?
Do the chaperonins also help denatured proteins get back together? Or
Do chaperonins help denatured proteins to get back together? Depending upon the nature of the denaturation, they can. But they're more likely to be involved in helping newly made proteins to come together. Okay? So, let's jump to the pre. I'm going to come back and talk about this other stuff later. Misfolded proteins were a very surprising answer to the cause of a disease known as mad cow disease. There's a human equivalent of it known as Creutzfeldt-Jakob syndrome. I think it's in your notes. Don't worry, I'm not going to spell it here. I'm not going to ask you to spell it. But a human disease that's very similar to that. This type of disease is found throughout the animal kingdom. It's found in sheep, where it's called scrapie. And every one of them involve basically dementia. The brain goes wacko, loses ability to function over time, and the organism literally loses its brain. Okay? Very, very severe problem. And for many years, people looked for the cause of this disease. We think of a disease, we think, wow, man, I got to find the virus. I got to find the bacterium. But no matter how much they looked, they couldn't find the virus or a bacterium associated with the disease mad cow disease. They couldn't find it. They looked everywhere. And finally somebody said, it's not caused by a virus or a bacterium. It's caused by an infectious protein. This was a radical leap in our understanding of this disease. An infectious protein. Okay? Such a thing had never been described before. The man that came up with this theory is known as Stanley Prusiner. He proposed this as a mechanism of how mad cow disease, actually I believe he was working on scrapie, but it's the same thing. Okay? He said this is how it was caused. They laughed at him. You're nuts. There's no such thing. How can a protein be infectious? We think of a virus as infectious because it injects its nucleic acid into cells, and the virus copies itself many times, makes many more viruses because there's genetic information there. How can a protein be infectious? We put a protein into a cell. What's it got to copy? It can't copy itself. We don't copy protein to protein. What happens? It turned out he was right. And what turned out to be the case was very, very surprising. The infectious protein turns out not to come from exogenous sources, although that can be a start. It comes from endogenous sources. Every person in here has the mad cow or the Creutzfeldt-Jakob protein in their brains. Every one of you in here has that. Okay? It's part of our brains. Our brain uses this protein. However, when this protein doesn't fold properly, it does literally what's shown here. And instead of going from a normal, nice, rounded shape here, it forms these blocks. And these blocks do something of association with each other that cause big globs in the brain. They literally destroy nerve cells. These are called plaques. A misfolded protein does that. Now, the interesting thing, and the reason we associate this with being infectious, is because once we get one misfolded one, it favors the misfolding of other ones. Why? It does. Okay? Her question is why. I want you guys to think about how that might happen. Okay? And the answer is not fully known, but from what we've talked about so far, you actually can have some ideas about how that might happen. Okay? So before next time, I'd like you to think about that, and I will discuss it at the beginning next time. Did you have a quick question? Okay, I'll say, ask me next time. Okay, see you guys later.
What was the word that you were using for that proteins know how to fold correctly?
Chaperones?
Yeah.
Chaperonin? Yeah.
Chaperoning?
Uh-huh. Yeah, it's in the notes.
Okay.
Yeah.
What did you say infectious proteins came from? What type of?
Your cells. Your cells code for them. Every organism has these proteins.
You said covalent bonds would connect the helix
Yep.
together. Wouldn't it be hydrogen bonds? Because isn't it?
They can be hydrogen bonds as well.
How do hydroxyls covalently bond though?
How do they? They split out water.
Oh, so they get rid of the hydrogens? Okay.
Well, hydrogen and one oxygen.
Oh, okay.
They split out water. So they're left with an oxygen bond in the place. Oops, sorry. Does that make sense? Clear as mud? Okay, good. If I get clearer than that, I start worrying.
It was like, how dare you?
Some lady. Hey, Thompson.
Saying congratulations.
This
So
one.
with the infectious protein, is it just something wrong in the amino sequence that like causes it not to fold correctly?
It's perfect. It's only the folding.
It's not.
You look at the sequence of a misfolded protein and the sequence of a regular protein, they're identical.
Is it by like chance or is it like?
Well, it's not fully known, as I said. So, but you can imagine there are maybe some things that will favor the misfolding of one to make another misfold. And that's what I want you guys to think about.
So is it kind of like Alzheimer's? Like.
It is, in fact, the disease manifests itself like Alzheimer's, although it's a different disease. It's actually a different problem than Alzheimer's. But the manifestation is very similar.
Okay.
Yeah. Scary.